Hello everyone, welcome back. Week 5 here, our second week of sauerkraut fermentation. I hope everything's going well. I have been looking at the discussion board posts and it looks like everything's going good on that first week, so hopefully it keeps going well for the majority of you. Now, just a quick continuation of what we were talking about last week. So we have slime-forming lactics on SGA that we would have been testing for in the lab, and then EMB that is testing for gram-negative bacteria, and coliforms, especially E. coli, are going to be showing up with this dark green sheen on EMB. So, what we would have been testing in the lab as well is the tomato juice gelatin, which can be used to differentiate between homofermentative and heterofermentative organisms. So, how this works is you have this porous tomato juice gelatin at the bottom that allows for gas to move through, and then you have a seal at the very top, and the seal is going to stop any gases from being released at the top of the test tube. So any gases that are released are going to go up through this gelatin, and then they're going to be trapped in between this gelatin and the seal. So you'll have a large gas bubble that appears if there's any gases produced. So with heterofermentative organisms, they produce CO2 as a byproduct, so they're going to be producing gas bubbles. So we can differentiate between heterofermentative, which has gas bubble, and homofermentative, which has no gas bubble. And then another thing that we'd be testing in the lab is milk agar plates, which form clear zones around the colony. These clear zones form around the colony if the organism that's growing there has proteolytic activity. So that basically they can break down proteins, they can lyse them, proteolytic. And so that's going to break down the proteins in this milk agar, and then you're going to be able to see a clear area around the colony. So if you see that clear zone, that means that this organism growing there has proteolytic activity. So for Lab 5, we're talking about gram-negative rods, specifically psychrotrophs. That is because psychrotrophs are mesophilic, so their optimal growth temperature is between 20 and 30. It's kind of your average temperature. But they do grow well at or below 7 degrees Celsius. So optimally is kind of room temperature, but they can grow at temperatures below 7 degrees Celsius, which is very atypical for most organisms. But that's what makes these psychrotrophs special. So the problem with them growing at these low temperatures is they can be responsible for a lot of food spoilage, especially in the refrigerator. So putting your food in the freezer will stop these from growing, but putting it in the refrigerator will not stop the organism from growing, unfortunately. That's going to make the psychrotrophs responsible for the majority of food spoilage that goes on inside the refrigerator. So there are a few pathogenic organisms, Yersinia enterocolytica, which will cause yersiniosis, Vibrio species will cause cholera, Listeria monocytogenes causes listeriosis, and then there are several that are spoilage organisms. We have, for the gram-negative rods, Pseudomonas and alkalogenes. And then for gram-positive rods, we have Bacillus, and gram-positive cocci, we have Enterococcus. So food spoilage organisms are Pseudomonas and Enterobacteriaceae. Right, so gram-negative rods can be any type of these three, intestinal pathogens or fecal contamination. So they're all, if you find gram-negative rods inside of your product, so you test it and you find a gram-negative rod, then that can be an indicator of any one of these things. Keep that in mind. So for the psychotroph gram-negative rods, which are again Pseudomonas and alkalogenes, they can be separated from the other gram-negative rods we're talking about because of their oxidase positive, proteolytic, and their non-fermentative. So these three characteristics are going to help us differentiate from the other gram-negative rods. With this graph right here, this little figure, if you follow it, so you have a gram-negative rod, and then you can follow these tests, and then depending on the results, that'll help you figure out exactly which organism you're dealing with. So if you found a gram-negative rod in your sample, and you performed an oxidase test first, If it was positive, then you'd know it's this organism, Pseudomonas aeruginosa, and if it was negative, then you'd need to perform some more tests in order to differentiate further. So if it was negative, then we could go McConkey agar, and then if it was red colonies, we'd go down here and then perform the indol test. If it's positive, it'd be E. coli. If it's negative, we perform the urease test, and then depending on if it's positive or negative, it'll be this or this organism. And same with the white colonies. So we can just keep going down and using these tests to further differentiate and figure out which organism we're dealing with. So as long as we know it's a gram-negative rod to begin with, as long as we follow this tree down, we can figure out exactly what organism it is. Our first organism that we would have been working with in the lab and taking a closer look at is Pseudomonas fluorescens. So an interesting characteristic of this organism is that it produces a yellow fluorescent pigment called fluorescein, and it's going to be found in soil and water, and it'll also produce a heat-stable lipases and proteases. So it's going to produce both of these, and so these enzymes are going to possibly survive pasteurization and continue to cause spoilage even after the milk has been pasteurized, which is going to make this a very important organism for food spoilage, and one of the reasons we talk about it. And the next organism is alcaligenes. So the specific species is Alcaligenes viscolactis, which can cause ropiness in milk, and that's due to the production of exopolysaccharides. And it's also going to be present in milk and spoiled eggs as well. And with that, that's all that we're going to be talking about today. Make sure to watch the 2 videos on discussion board and leave your on the Blackboard, and then leave your discussion board post after taking your quiz. Good luck, everyone. I hope that everything is going well with your sauerkraut, and if you have any questions, feel free to let me know. Goodbye.